this program, and he has been here. That is, all of us should understand that what importance he assigns. He is the chief architect of this universe. Therefore, it is told as uh, please. He is the architect of Amity Universe, where this has great campuses all over the country and internationally. And as you can understand, the type of facilities which have been created all over, there are so many parallel symposia organizations going on in this campus. And not only that, and because of that, you will find since morning so many people, and all these 5 days, so many people have been coming to talk to you people. It is not easy just that people will spend time to tell briefly, just in one line, not as a credential, what other things will be introduced to you. But Professor Ray, he had an appointment with the UGC chairman this morning, and he quickly finished that because he had to come, and he wanted to be here with you. So this is what is a commitment for the students that with people have. And I'm so grateful on one hand to those founder, president, chancellor, vice chancellor who has created this and allowed this. On the other hand, the other link is Professor Ray and others who have been talking and coming to talk to you. With these brief words, with expressing my gratitude to each of the speakers and to the establishment, now I leave it and hand it over to the anchors, and who will start with. Okay, please.
So now it's time to introduce our next speaker, Professor Krishanu Ray, who is going to give the valedictory session of the day. Professor Ray, Director, National Brain Research Center, Manesar, formerly Professor and Chairperson, Department of Biological Sciences, Tata Institute of Fundamental Research, Mumbai, is very prominent figure in science with focus on understanding the logistics of movement of the biological motors to regulate every possible movement at the cellular and molecular level in appropriate biological context using Drosophila melanogaster as a model system. He has identified the molecular basis of cargo motor interactions involving the tail domains of a unique heterotrimeric motor protein called kinesin 2, and is in the process of identifying the use of this interaction in establishing the synaptic connectivity in the larval ganglion. We are also engaged in studying the effects of sensory stimulation on intracellular transport carried by kinesin 2. With over 100 publications, numerous citations, and fellow of all science academic in India, his work and dedication has not only improved our understandings about axonal transport, cytoskeletal dynamics, and spermatogenesis, but also inspired us as students. We are honored by your presence amongst us, and we eagerly anticipate the stimulating exchange of scientific ideas and knowledge that your participation will undoubtedly encourage. So, without taking much time, I request Professor Ray to please shed light on his work and enlighten us all. So, the topic of his session is studying axonal transport and cellulogenesis in Drosophila, correlating molecules to both cellular and behavioral attributes. So, we welcome you here.
Okay, thank you very much for a very generous introduction. I'm not sure whether I really fit the bills, but thank you. Uh, so, who all here are undergraduate students? Come in the front row. All the undergraduate students. Because this lecture is for you. Yeah, please. There are lots of seats here. I'm really happy that you came. Yeah, I prepared this for you only. Okay, and feel free to interrupt me. Yeah, from the first word that I utter, and I will not be. I'll be very happy to answer your question rather than saying what I'm supposed to say. Okay, so what I decided to tell you today is how protein molecules and simple chemicals like lipids kind of interact with each other and create a chemical intelligence, which is as astounding as the intelligence that your brain can produce. So I'll tell you about a traffic problem that all our cells solve every second, as we speak, in all our body, in all our cells. And this traffic logistics that needs to be solved on a real time basis is the hallmark of life. So, in other words, if you want to define life, the simple word of life is motion. Okay. So we recognize a living object by its motion, because other than the man-made artifacts that like the automobiles and aeroplanes, the most of the living objects in real world are animate objects. Yeah, that's the word for life, animate. So therefore, the motion signifies life at every scale. So today I will talk about the motion that happens at the nanometer scale. Yeah? Do you have any clarity on what is a nanometer? 10 to the power ninth of a meter, right? So in that scale, the movement happens, and this movement happens on a step by step basis. Each step takes about 8 nanometer. Hmm? Now the distances that needs to be managed by taking this 8 nanometer step is huge. Yeah? So some of the neurons inside this hood is about 1000 meters long, a single cell. Yeah? That's an estimate, not a real number. But the real numbers are also quite long, because your sciatic nerve is a single neuron, right? A motor neuron that starts from the lumbar and ends at the toe is a meter long. So think of a blue whale and think of a 30-meter long animal. What will be the length of the sciatic nerve? So these are the real distances, right? Now, how will you cover a distance of hundreds of meters using 8 nanometer step at a time? It's like walking around the earth, right? But we still did that, right? Humans have spread around the earth simply by walking. But that took a long time. But here, the challenge is that it has to be solved in real time. So as I speak, the synapses in my larynx, the muscles that control my larynx, is active, right? And these synapses muscles are constantly, contracting and expanding. So to manage that, this neuron and the muscle is constantly talking to each other. The neuron and the way, how do they talk? The neuron sends a chemical out called neurotransmitter, and the muscles receives that chemical and makes sense out of it. Yeah? So what the muscle does is that when it receives the chemical, it releases calcium inside the muscle, and that calcium will contract the muscle, and that as a result my muscles will constantly contract and expand depending on the rate at which these chemicals are thrown at this muscle. Now what is there at the base of this muscle? A protein called myosin, yeah, a motor protein. So myosin gets activated upon calcium stimulation. How? Because there is another protein, which is the myosin light chain kinase, which will put a phosphate onto a certain amino acid on this myosin motor. So once this amino acid is phosphorylated, its charge is changed to something negative. And that little change in charge causes this motor to hydrolyze energy quotient called ATP, extract the chemical free energy of this ATP, and use that chemical free energy to generate a mechanical force. exactly like an automotive engine. An internal combustion engine of an automotive does exactly the same thing. It burns the fuel, extracts the chemical energy, and uses that chemical energy or thermodynamic energy to make a work, yeah, to produce a force, and that force ultimately drives the work. That's the physics of it. The molecular motors are the smallest internal combustion engine ever made in this universe. So these motors, like myosin, have the ability to extract this chemical free energy in a very, very efficient manner. The efficiency is mind boggling. We haven't made such a machine ever. So it's almost 40% efficient. And it and the fastest Ferrari engine is 29% efficient. So you can imagine, how well oiled these machines are. They extract this free energy and produce a massive force of 5 piconewton. You will, if you do the ratio, you will realize how this 40 kilocalorie per mole energy can generate such a huge force. The force conversion efficiency is 93%. Yeah? If we had ever built such a machine, we would reach moon in no time. Yeah. So this is life. So today, what I'm going to tell you, unfortunately I'll only tell you about axonal transport. I don't think if I keep talking about everything, you will be here till dinner. So I'll only talk about axonal transport, and I'll tell you how this particular movement has a bearing on understanding how the brain functions. Yeah. So I'll make a long connection from the brain, which is a huge macroscopic object, to a motor protein, which is a nanoscopic object. Yeah. In next 30 minutes. Yeah. So, okay. So hold on tight and we will start. Now, this is a picture of a human brain, and this is given by my colleague Arpan Banerjee. And this is, what you see here in color code is what is called the DTI map of the brain. Okay, so this is a tensor map of the brain, which is generated using the NMR signal that the water molecule generates. So using that water molecule signal, we can actually produce the connectomic map of the brain. We can see from where to where the neurons are connecting. So essentially what you see is the major connections in the brain. These are the highways of the brain, or the electrical trunk line or communication cables inside the brain, whichever way you can think of. Now, to understand how the brain functions, we need much more than understanding how the brain is connected. That's the first step. But the brain functions are, has a multiple different, aspects. Behavior is obviously the first and most important aspect. The structure, as I showed you here, is another very critical aspect. To understand how the behavior manifests, we need to know how the brain is structured. For example, you are listening to me, right? So what is happening inside your brain? So first thing is that your ears and your eyes are sending electrical signals. Those electrical signals go to different parts of this huge mega structure, right? So the eyes talk to the visual cortex, the ears talk to the auditory cortex, and they're close to each other. They're miles apart. So now, how will these two, different parts of the brain have to converge in some other parts of the brain to make sense of what you are seeing and what you are listening, and also make sense of what you are listening? This abstraction of language is the creation of our brain. Okay. Of course, many other species also have a language, the birds particularly, but the humans are the only animal which has the most nuanced and highly sophisticated, grammar and language. It's created by our brain, and it's created by the neuronal network in our brain, right? And the neurons are like computer chips, so they, create these processing circuits, and the circuits talk to each other. So therefore, you need a physical or electrical connection between this auditory cortex and the visual cortex somewhere else, plus in between, lots of other abstract, processing to create what you will describe as a feeling, right? You are either feeling good or bad, or satisfied or dissatisfied, or understood or not understood, bored or excited. So all these feelings are essentially a handiwork of this neural network. Yeah. And this neural network is nothing but an electrochemical computing system. So this electrochemical computer, which is our brain, therefore, and the chemistry always works on contact, right? An enzyme can only catalyze the substrate if the enzyme molecule and the substrate molecule are physically attached to each other. An enzyme cannot catalyze a substrate at a distance. So chemistry happens on contact, electrical communication can happen at a distance, and the brain combines the two to generate this enormous, humongous computing power, which is actually much better than the best supercomputer we still haven't built, right? Now, to understand this brain function, you need to understand it at a multiple different paradigms. Yeah? So, for example, the behavior, which I already talked about, there are two aspects, innate and learned. The innate behavior is what controls us, like our heart, our internal organ functions. So, these are all innate. The brain, you don't have to think to pump the heart, you don't have to think to breathe, you don't have to think to control your breathing as you are talking. So, these are innate behavior of our brain. It controls the system innately, which are very, very complex if you think deep enough. But then there are learned behavior also. For example, you are sitting here because you have been taught to sit here and stay quiet, right? When you are 2-year-old or 5-year-old, you would not sit here quietly. You will be very cantankerous and asking lots of questions or doing something totally different. And then gradually your brain network has been streamlined, and lots of synapses are burnt and a lot of new synapses are formed and a lot of new neurites grown, connections made, and your computer is completely destroyed and rebuilt by the time you reach here. So you have now a new brain, and so that's the learned behavior. Everything, and this learning is a constant process, and it will continue forever in your brain. So that's another aspect of the behavior. Then the physical connections between this, and the word for it is called connectome because we assign the connection to a behavior. That's the connectome. Otherwise, it's just a connection. Okay. Then the functional networks, which is very important to understand disorder and diseases. You have a stroke, and the physicians looks at your MRI image of your brain and tells you, oh, this part of the brain is dead. So that kind of aspects is also very critical. Then the cellular and molecular aspects. So you have, as you learn, you grow more neurites in your dentate gyrus. You have these Purkinje cells which have very highly branched neurons. So those aspects are also needs to be understood to understand the brain function. And then this input output correlation of a neuron, which is the physiology of the process, etc., etc. Okay. So this is one example of how one can study the brain function. So this is called electroencephalogram, which is simply recording the electrical field potentials that is there surrounding our head. So all your heads is emitting a lot of electromagnetic signal. So it's like a radio, which is, or a radio transmitter, which is sending signals. And because the brain is computing in real time, a lot of electrical activity is going on, so it generates the electromagnetic signal like a radio wave. And you can record it by putting an electrode around the skull, and you can get this kind of frequencies which are kind of, this is the sort of, you record this complex signal and then you kind of dissociate them and you break them down into fundamental oscillations, and you get this what we call alpha, beta, gamma, theta, delta oscillations. And they signify different aspects of our brain function, which is written here. The perception and consciousness is attributed to the gamma and beta functions. The alpha is active, like right now, if you take an EEG of my head, you will see a very strong alpha signal, high powered alpha signal. Attention, awareness, memory consolidation. When we go to sleep, the alpha, beta, etc. quiets down and the theta rhythm gets more prominent. It is there, but you cannot see it when the other rhythms are there because it's a low frequency signal, and so on and so on. Okay. So now we have to make sense of all these signals that is coming out, which is kind of a soup of information uh, to an individual. For example, suppose I want to know, as I am speaking and as you are listening, how your visual cortex, what is the rhythm with which your visual cortex and the frontotemporal lobe is communicating with each other, because that's where the signal is understood actually, right? So what kind of rhythm is there between your frontotemporal lobes and your auditory cortex? And how does it change when you are intoxicated with something, or when you are satiated, or when you are disturbed, stressed, or when you are running, or when you are sleeping? So there are so many different modulations that can affect this interconnection, or crosstalk between these various centers of the brain. And how will we know that? By just taking a soup of information. It's like recording all the internet chatter in one place and trying to make sense about the communication between you and me through the WhatsApp, right? Imagine that. So that's what these EEG signals are. And that's the challenge that the scientists have. And the problem is called source localization problem. And it has no one solution, and it is still not properly solved. Yeah. So a lot of things are still going on. And then, there are this neuronal anatomy that you need to solve. You need to see these neurons, you need to see their connections. And this is a typical, neuron that you see here. For example, here, this is the, mouse brain stained with Golgi staining. And these neurons make contact with synapses. And these are the sort of dendritic ends, the red mark at the places where the neurons and dendrite and the axons and dendrite talk to each other. So, and this is how it looks. You see this dark patch. This is the electron micrograph, and you see these vesicles. These are the filled with neurotransmitter. They release the vesicle as soon as an electrical impulse reaches the synapse, and then this is the receiving synapse, post synapse, and it receives the signal, and you can see that it receives signals from two different neurons. And now it has to disentangle that signal. It has to learn which neuron has talked which way to make sense out of those signals. So these are the signal integration that happens at the postsynaptic neuron. Okay. So now if you compare this whole processing activity, and so this is a comparison, size scale comparison and energy scale comparison. So as you see, the fastest supercomputer that we built is about 30 quadrillion bytes, and these are the number of LEDs that is equivalent put in here, some 93 million LEDs. Yeah? This is our brain, which is much smaller compared to the best supercomputer in terms of the byte processing capacity. But look at the energy. So this requires 9.9 million watts, which means you have to put up a nuclear power plant to run it. This requires only 20 watts. Yeah? So that's the efficiency of the brain. It's energy efficient. It's the most energy efficient supercomputer that we have. And then these are the cat brain, and this is human genome in comparison. This is your iPad, and so on. So obviously, you can see that the human brain is a humongous beast, and it's also one of the most efficient beast. Okay. So this is a Drosophila brain. Now, obviously, solving all the connections, all the neurons, all activity in human brain is still beyond our reach. Maybe one day, in your time, you will know it, or you will discover something very fundamental in your lifetime. But we can't understand the human brain just like cutting off the brain and putting an electrode into the brain. It's not so simple. Okay, we need to do a stepwise work. So instead of 35 million neurons, how about 3,000, 4,000 neurons? That's much more manageable, right? So that's Drosophila. So we can easily use Drosophila with a few tens of thousand neurons, then, and which are nicely segmented like a human brain. There are each color shows different functional centers of the fly brain. For example, this is the visual cortex equivalent, right? So this is where the visual signal comes from the eye. This is the olfactory lobe of Drosophila, which is the equivalent to the olfactory bulb of human brain. And this is the central complex, which is like the cerebrum of our brain, which controls, this is the mushroom body, which is equivalent to the hippocampus of our brain, where, things are learned, and so on. Now, if we look at this center, this mushroom body center, you can see this is a single neuron. And Drosophila is the most complex animal for which now we have the full connectomics map. We know every neuron, we know every synapse, we know everything that they connect to, and we know their functional correlation. So this is the most visible, structurally and functionally most visible complex neural architecture that we have learned so far, and we are going to learn more in the near future, and more advanced animals like mice. But here in Drosophila brain, you can now see, so these kind of extractions you can do. There is a database called this codex.flyaware.ai, and this, you can go to this database and all this data will be available to you. And there is a lot of things to do using this data. You can start your own research in finding out connectomics in Drosophila brain by analyzing this database. This is a raw data, is still not fully analyzed. Okay, but this is one kind of analysis that you can see. There are neurons which connect to the central complex, which is the cerebrum equivalent, which controls the motor activity in the fly. This is the human brain network, human neuronal network. So now I will switch gear and I'll come to a single neuron. Okay, so a single neuron is a very long cell, and the cell, all the library or sort of say database of the cell, so to say, is kept here in the nucleus. That's the DNA where the database is kept. Now, this database or the protocol book or the manual of neuron has to be read every now and then, and the message has to be extracted in chemical form, and then that message has to come out and reach here for the neuron to function. Okay, and also there. Now, if you mix up this message, if you send the message which is supposed to be here on this side, will the neuron be able to function? Obviously, no, right? So that means this particular cell has another logistical challenge to make sure that it sends the right message to the right destination. And as I told you, the destination sometime can be very, very long. So this is the, I have already spoke about all these things, so I'll skip that. I'll come to the synapse. Now, synapse is not just an electrical junction box. Synapse is also an integrated circuit processor equivalent. So how does it work? So in case of synapse, when an electrical signal is reached, it releases calcium from the internal store. And when it releases calcium, lots of things starts unfolding. It's like a domino. You just flip one and then starts chain reaction. This is one such chain reaction, which is the release of the neurotransmitter. Okay, the names doesn't matter. What matters for you to remember is that this particular function alone requires about 400 molecules, 400 different kind of molecules rather. So this area of this brain has to have at least 1,000 different kind of molecules in right proportion and in right condition to enact this process as the electrical signal reaches. If it did not, I would not be talking so coherently, and you would not be sitting and listening. You will fall off the chair. Okay, so that is the efficiency of this system, and that is the logistics that has to be managed. Be it protein, be it lipid, be it RNA, or be it calcium, all these requires to come from the cell body mostly, right? and therefore the neuron has to maintain a supply chain which should not be like this bridge that I crossed today, which took me 10 minutes. Yeah, it should not be this the national highway between Gurgaon and Delhi, which took me 1 hour to traverse, and with unpredictable traffic jam. This neuron will not function, right? So, so this is the logistical challenge of a neuron. It's a long axon, sometimes hundreds of meters long. The only source is nucleus, and all these things has to come. So therefore, the supply chain must keep up with the consumption. If I am running, the synapses in my leg muscles and other body muscles are constantly firing. So, which means that every enzyme has a limited life cycle. It can work for 50 cycle, 100 cycle, 1000 cycle, or million cycle. But at some point, it will give up. Yeah, the thermodynamics will compel this enzyme to stop because it will generate a lot of mechanical heat inside the structure, which will melt the structure. The protein will misfold or unfold. So enzyme will stop functioning. So therefore, you need to constantly replenish this enzyme. The more you active, more your muscles are active, more enzymes are needed at the presynaptic complex and as well as in many other places. So this is the logistics is happening in real time. And there is no central comparator to direct this traffic. There is no Professor Malik sitting at the back and making sure that all of you listen to me in an attentive manner. Nobody. It's happening autonomously. Okay. If it doesn't happen, then you have a problem. So there is this network of activity between these neurons which coordinates with each other, and when it breaks down, the neurons die and we forget. That's the disease called dementia, or Alzheimer's, Parkinson's, Huntington. All these things happen because of sorry, I think my slides are going ahead of me. All these things happening because of this traffic congestion in either the axon or in the dendrite, mostly in the axon. Yeah. So all these diseases begin essentially with a traffic congestion. And this traffic congestion happens because of miscommunication between different parts of the system. Okay. So then how do we, so therefore to understand that, we have to understand what is this traffic. Now this is an example of an innate traffic inside the cell. Okay. This is something that you are all familiar with. You have read in the textbook that the proteins are, synthesized in the endoplasmic reticulum. They are glycosylated in Golgi and then they are sent to the plasma membrane. This is how it happens. Okay. What you see here is a green fluorescent protein tagged to a viral, surface protein, glycoprotein, which is making a beeline to the Golgi as soon as it is made. And how do we know? Because the protein is fluorescent. So we can see that as soon as the protein is made, it knows where to go. And this is the intelligence that I'm talking about. How does this protein know where to go? How does the cell know where to send it? How is it getting directed altogether? So this is the innate traffic inside the cell, and it never stops. Okay. This is random motion. So initially people thought that this is random motion. But if you look at this random motion, this is just carbon nanoparticles floating in water. Okay. And if you look at this motion, you will immediately recognize that this is no random motion. Yeah. This is very, very orchestrated, organized motion. If it was a random motion, then it would have been diffused all over. The fluorescence will be uniform all over, but it is not. Yeah? So this happens because of the motor protein, and this was discovered from a model organism called squid. So squid was used as a model to understand neurophysiology because it had this giant axon. Okay, this is the giant axon of squid, motor axon, and this controls the swimming behavior of this animal. Have you seen squid? This is what it looks like. It also tastes very nice. Yeah. Greeks call it calamari. And so this is this, the long giant axon. And so this is used by, neurophysiologists since Hodgkin and Huxley first discovered that the neurons fire electrical signals. People have been using this as a celebrated animal for electrophysiology. But One young postdoc, Nobutaka Hirokawa, in Shina Inoi's lab, thought otherwise. So thought that, okay, why not crack open this neuron and axon and see what is there inside. So he developed a very innovative electron microscope technique called deep H fridge fracture scanning electron replica based scanning electron microscopy. It's no longer in practice anymore, but it used to be the most important the microscopic tool to see what is there inside once upon a time. And when he did that, he found that inside the neuron you have these large vesicles, and there are these long tubules which were that time known as microtubule, and these are the microtubule. And you see these bridges, cross bridges connecting this large structure to this microtubule. So this raised the first question about what is this structure. Now another undergraduate student, Ron Well, who was acting as a the assistant in a microscopic course taught by Shina Inoi at MBL in Massachusetts, long time back in 1985, 1982 actually. And a young postdoc called Scott Brady teamed up together to also find these cross bridges for a different reason. So they started purifying proteins from the squid giant axon and started looking for their microtubule dependent ATPase activity. And the Scott and Ron both succeeded in a different way, and they purified this protein. But Ron went there first. Yeah. So Ron Vale is a young undergraduate student, published the first paper describing this cross bridge and called it kinesin. And that's how the journey began in 1985. There was this landmark paper published in the journal called Cell. In 5 papers came out in 5 months subsequently. And those 5 papers changed the course of our understanding of intracellular transport, and done by an undergraduate like you. Yeah. So these kinesins can be purified. You can see them under the microscope, and you can also attach them to plastic beads, and you can see and put them on microtubule, and they will start moving in a step by step fashion without stopping. So kinesin is one of the most resilient and tenacious motor which will keep on working as long as the ATP is available. And these motors, now we understand how it produces the force. It hydrolyzes the ATP. It binds to this particular subunit on microtubule. Microtubule is a polymer of alpha and beta tubulin, which is like Lego bricks join each other head to tail and makes a long chain. And 11 or 13 such chain join side by side and create a tube. And that's called microtubule, which is about 25 nanometer in cross section. And a motor protein binds to only the beta tubulin subunit of one protofilament. So that means you can think of microtubule is a 11 lane highway, expressway. And the motors have a very strict lane discipline, and they walk on this beta tubulin subunit on each lane, step by step, one hand over hand, as it's shown here, and cover this distance from the nucleus to the synapse. Okay. This is the simulation, and you can also see them. This is the single molecule data. The red is microtubule, and the green dot is a kinesin motor. And you can also see this motion at a single molecule level. This is done using the total internal reflection microscopy. And you can also plot the step cycle like these, and you can see them going step by step by step. This is not my data. I forgot to put the there is the citation. Okay, so in the axon, so the humans make 35 different motors. Yeast makes 7 different motors. Drosophila makes 25 different motors. So each animal has their own cluster of motors. The 7 motors that the yeast makes are mostly actually entirely dependent on segregating the chromosome. Okay, And the humans have again some 17 different kind of motors segregating the chromosome, because human chromosome is a large complex structure. But in addition to that, the eukaryotes from Drosophila to humans, in fact from C. elegans, from C. Chlamydomonas to humans, make other motors which are not required for chromosome segregation. So those are the motors which is required for the intracellular transport. And these are numbered 1, 2, and 3. Okay, and they are highly conserved. You find them in every organism which has an extended cell, all eukaryotic organisms. And in the axon, these 3 kinesins move towards the synapse, and there is a humongous beast known as dynein, which looks very different from the kinesin, and the independent family, superfamily, and that moves from the synapse to the cell body. And this is the traffic motor of the cell, locomotives of the cell. And I already showed you kinesin. This is how the dynein looks like. It's a huge structure, and they have 4 legs. They walk on this microtubule, and they have the ability to go from one lane to the other, and that helps them enormously and to navigate through the cell, because these microtubule highways are not like our highways. They are littered with cows ruminating on them. Those are called microtubule associated proteins, or MAPs. They decorate the microtubule. So when a motor has to walk on this microtubule, it has to be mindful of these MAPs. It has to navigate, tiptoe against around the cows that are ruminating on this highway at a breakneck speed. Yeah. So that is another marvel of chemical engineering inside the cell. Now we study this movement using Drosophila as a model, and this is the sciatic nerve equivalent of Drosophila, for example, and these are the sensory neurons that you are seeing here. So this is the ventral ganglion where the synapses are, which is equivalent to the spinal cord, not exactly, but somewhat. Yeah. And these nerves combine both motor axons and the sensory axons. Here you are only looking at motor sensory axons. In the larva, which looks something like this, the fly is about 3 millimeters long. The larva is also similar in length, and these axons can be as long as 2 millimeters. That is the longest of these axons, but on an average about a millimeter length. So we study all the movements using these axons, and this is how it looks like. And this is the and these axons make synapses. This is a typical synapse that you can see here, and and these synapses have very you can also see the composition of these synapses. For example, the green here is an enzyme called cholinestyltransferase, which makes the neurotransmitter called acetylcholine, which is what our muscles need to contract. And the N-acetylcholine receptor, nicotinic acetylcholine receptor, which is in red. And as you can see, the acetylcholine is in the presynaptic compartment, and the receptors are on the postsynaptic compartment. So using the super resolution images, you can actually distinguish them. Now, what we wanted to know is how does this cholinestyltransferase reach the synapse? Why? Because in autopsies of patients who have unfortunately died in Alzheimer's, it was shown that the one of the cause of the Alzheimer's is a reduction in the cholinergic activity. Okay, so much so that people are given a drug called donepezil or others, which are inhibitors of an enzyme called acetylcholinesterase, which cuts off acetylcholine and cuts off the signal. So if you prevent this enzyme to act, then your signal will remain longer, and as a consequence, it was shown that you will be developing Alzheimer's at a later stage. So it was a prophylactic therapy to prevent an onset of Alzheimer's. And it was also shown that cholinestyltransferase levels reduce significantly in the dentate gyrus of the hippocampus, which is the seat of your learning and memory. So therefore, understanding how cholinestyltransferase or acetylcholinesterase for that matter reaches the synapse is very critical. Now both these enzymes are actually synthesized in the cell body of the neuron. and then transported to the synapse. Now, a lot of people thought that it can simply diffuse from the cell body to synapse because it's a soluble enzyme. And that theory persisted till I published my first paper. Okay, and that was 25 years back. So I did not realize, but we realized together that this movement of cholinesterol transferase cannot be concluded using diffusion alone. So we devised an experiment, something like this. This is the most refined form of this experiment. It was not like these 25 years back, but what we did was we made cholinesterol transferase coupled with a photoactivable green fluorescent protein, and also coupled with an red fluorescent protein called mCherry. And then we activated this enzyme in the cell body using a laser light. And once we activated it using the laser light, the green violet light, it started fluorescing in green, and this is what you see. It still looks like diffusion, right? It still looks like, it's just flowing like water. But when we made the chymograph of this movement at different stages of the development, we found a difference. For example, at this stage, which is 78 hours after the fertilization or egg laying, whatever you call it, this flow was maximum. But later, it comes down again. So this is the chymograph data. Okay. So this suggested that this must be regulated. So the question is how? So to cut a long story short, we found out that there is a motor called kinesin 2, which is required for this movement. Okay. And kinesin 2 is like kinesin 1, is a heterodimeric motor protein. And we found that kinesin 2 can directly bind to the cholinesterol transferase. So in other words, kinesin 2 seems to bind to each enzyme and carrying them to the synapse, the most inefficient way to make any transport, in my opinion. But still it happened that way. Okay. And so I'm not showing you the data. And we also learned that the activity at the synapse is very critical. If we inhibit the cholinergic activity by putting a drug called hemicolinium, which prevents the recycling of choline and acetylcholine synthesis, you see this movement is completely abolished. Okay, so therefore it cannot be diffusion. And we also found that this motor kinesin, we used an assay called fluorescence resonance energy transfer assay, or FRET. In simple terms, if you know fluorescence, you all know fluorescence. If you don't know, I will explain. Okay, I'll explain anyway. So fluorescence is very simple. You shine a UV light, it gives you red light or green light. Okay, and you have used it in lots of parties. You put your UV paint and go to the party and look very snazzy. But this is also very useful because if you have two kinds of fluorophore, one that emits green and one that emits red and absorbs green, if you bring them very close to each other, less than some 7-8 nanometers, then instead of emitting green, this fluorophore, which absorbs violet or blue, will transfer the energy to the red fluorophore, which absorbs in green non-radiatively, and then this red fluorophore will emit red. So this is used as a nanoscale molecular ruler to understand inside the cell how far two proteins are in real time. Yeah, that's why this technique is very useful. And it is also useful to understand how a molecule functions. So this is one of the greatest tool discovered in chemistry or photochemistry that revolutionized our understanding of chemical enzymes. But we can also we use this tool simply to find out whether cholinesterol transferase can bind to this kinesin 2 motor. And when does it bind more? So this is the data. So as you see, the fluorescence intensity goes up with time of this motor of the cargo, and at this particular stage at 78 hours when the flow is highest, we see an increased binding with the motor. And this can be again abolished by putting the drug hemicolinium. So that's the black. Okay. So that's how we found that this motor can directly bind. I'll again cut the long story short to tell you that later on we used a physical modeling technique called hidden Markov model and simulations. And using that, we found that there is a combination of diffusion and active movement that propels this flow. Okay, so that biophysics is also very interesting, and that is because these axons are very narrow, and so these are generally a single file movement, and that's why a little kick by the motor makes it run very fast. It's like in a bowling alley, you just throw the ball on a slippery surface, and the ball moves very fast, right, after that initial kick, and it is exactly similar. Hmm. Now I will move to a different problem, and this problem is the movement of vesicles inside the axon. So what you see here are the lipid-bound vesicles. These are inverted color, so color is not important. This is a black and white movie, and this and each vesicle is moving through the axon. You see this white dot moving persistently. So sometimes these vesicles can move very long distance. The length of this is about 80 nanometer. Okay. So to cover this 80 nanometer distance at 8 nanometer per step, how many steps have to be taken? 80 microns, sorry. How many steps have to be taken? So 80 micron is 80,000 nanometers, right? So you have to take 10,000 steps. Yeah. As they say, 10,000 steps a day will keep your stress away. So these motors move very fast. And they move through this very crowded, so this is the sensory axon. They go through this very crowded space, which is also littered with mitochondria. So it's very congested tunnel through which this move. Yeah. And one can track this movement, for example, this way. So you can see this bidirectional movement by tracking signal. So not all movements are processive. Okay. So we asked that what triggers the start and finish of this movement, what starts them to move and what makes them stop, and what makes them change direction. So we don't have the full answer. So the question that we asked is, how does it start and stop, which is critical for understanding how this motor finds its address. Because if it's moving back and forth, how will it really reach where it has to reach? So this is a case of chemical intelligence. Now it happens at multiple level. So the motor doesn't really have to know where it has to deliver the cargo. The cargo doesn't have to know where it has to go. Only the place where the cargo has to be delivered must know where the cargo has to be delivered. Okay, it's like this. You keep running through the streets, and then people in the building are there with a hook, right? And when they find the right one, they hook you up. So this is exactly how the cargoes find their destination when they reach the destination. They're just dislodged from the motor using some chemistry. And this can happen at multiple levels. It can happen by disabling the motor. It can happen by disassembling the motor. It can also happen by dissociating the cargo from the motor. In case of cholinesterase transferase, it is the last case. The cargo falls off. In case of many other vesicles, also it happens. For example, the APP containing vesicles, which is the major ingredient for Alzheimer's. If APP vesicles derail in the mid course, then APP comes in contact with an enzyme called synuclein, which is a protease that will chop off the APP to A beta, and then A beta will start aggregating. And that's how the Alzheimer's starts. Okay. Now, APP kinesin interaction is regulated by phosphorylation. The APP carrying vesicles bind to the kinesin through this subunit called light chain when the light chain is phosphorylated by a certain enzyme called GSK3. So that phosphorylation causes the APP vesicles to bind, and if the GSK3 beta is inactive, then APP will fall off this or if some phosphatases come and dislodge the phosphate like PP2A, it will fall off. So there are many such ways. For example, this polyQ expanded repeats, which is the cause of disease like Huntington, or spinobulbar, SBMA, etc. That also acts through this phosphorylation. APP I just talked about, and the Parkinson's, which is caused due to this enzyme called parkin, which is a protease that dislodges mitochondria from the motor. So these are the various ways the cargo can be dislodged. So it was shown that GSK3 beta is concentrated at the synapse. So as a result, when the cargo moves and reaches the synapse, it will phosphorylate the motor and disengage the cargo. But if GSK3 beta is dislodged and distributed along the axon, then you will get Alzheimer's. Yeah? So who puts GSK3 beta at the synapse? That's the another question. So it is like that, one domino after the other. So same thing happens with kinesin 2. I'll just skip it. Now I'll come to the focus of the last part of my talk. So now the another way you can also hook up the motor to the cargo, also using phosphate, but not on the motor, but on the lipids on the vesicle. And in this context, this particular lipid known as phosphatidylinositol, which looks something like these, it has a sugar glucose moiety bound to this long chain aliphatic carbohydrate through a glycol. So these phosphatidylinositol are phosphorylated. There are these three positions, 3, 4, and 5. The phosphatidylinositol is phosphorylated into these three positions. And because these three positions can be permuted in different combinations, and you can also combine different permuted phosphatidylinositol in different combinations like 3, 4, 3, 4, 5, 3, 5, 5, 4, etc., etc. This has become the evolution's choice of pin code. So this is the chemical pin code that the cell uses to define an address. Okay, as you can see here. So each part of the cell has a particular phosphatidylinositol code, and the motors can recognize this code. Yeah, I'll show you how. And there are these enzymes called PI3 kinase, which makes this code. PI4-5 kinase, 4 kinase, 5 kinase, etc. And there are phosphatases also, which can make this code. Now, before I tell you that, I have to take another detour. I have to tell you about insulin, which is again a very important hormone in our body, a growth hormone also. Okay, and it is also very critical for not having diabetes. Yeah, and eating all that sugar and able to digest it. Now, insulin signaling in the lipid cells called adipocytes or fat cells is known to stimulate movement of RAF4 vesicles, and through RAF4 vesicles it is known to recycle GLUT4 receptor, which requires for taking in glucose inside the cell. So when you eat a heavy meal, your pancreas secretes insulin and it goes through the bloodstream and it stimulates the adipocytes to take up more glucose and burn them. Okay. Insulin signaling in the neuron is known to stimulate the axonal transport. Okay. And it is known to the transport recycle receptors at the synapse, and it is known to activate the movement of APP in the axons. The insulin signaling or lack of it in the brain is also involved in inducing early onset dementia. So Alzheimer's is also known as type 3 diabetes, because people who have overactive insulin receptor in the dentate gyrus gradually develops this dementia. Okay. So now, and we found that the RAF4 transport in the Drosophila is detrimental for maintaining the synapse. So in other words, if you put more RAF4 towards the synapse, you get less synapse. The synapses degenerate. Now, what is this connection between these three things? So to understand that, Kamal, who was a PhD student, who is still a PhD student in my lab, decided to throw some insulin onto the axon and look at the movement of RAF4 vesicle. So what you see here is that this is without insulin right here, and this is with insulin. So if you look carefully, you will realize that in presence of insulin, the vesicles are moving faster. right? As if they are on adrenaline. But these RAF4 vesicles are known to move on a motor called kinesin 2, which is actually a very inefficient motor. It moves very slowly at the rate of 0.5 micron per second. So how does insulin make this motor stimulate on adrenaline and run faster? That was the question we asked. And we also looked at this chymograph, etc., to prove that it is indeed due to insulin. Now, again, after a lot of work, we found, and Simanti, who was a MSc student in the lab, did this experiment, that insulin stimulation increases the level of phosphatidyl 3 inositol, PI3, on these RAF4 vesicles. This is the data. Okay, as you see, more insulin, more PI3 phosphate on this vesicle. And I'll cut this out again. Yeah, and we found that this green and red vesicles also move together. Now there is a kinesin called KIF16B, which is known to bind PI3 phosphate. So obviously then we thought that it may not be kinesin 2, it may be kinase KIF16B equivalent. And indeed we found that this KIF16B binds to these vesicles through PI3P, and PI3P is made on this vesicle by this protein called VPS34, which is a PI3 kinase, and because of that, these vesicles move faster. Okay, so that's the end of the story. So now, in summary, what I told you is that kinesin 2 motor is very versatile, and it can bind to different cargos in the same axon, transport them in at different levels. As you have seen, that CHA transport kinetics is very different than the RAP4 transport kinetics, and this transport is very critical for maintaining the synaptic homeostasis. Okay, and this is the data. You see, activated RAP4, there is very little synapse. Inactivated RAP4, more synapse. So there is a direct correlation between putting RAP4 at the synapse. And that has a behavioral correlate also. The larva learns to walk straight. And that is important for the larva to walk straight, to come out of the food, so that the pupa can form in a dry place. And when the fly emerges from this pupa, it will not be drowned in the soft agar food, right, or somewhere else. So that's the significance. And this was the most interesting data found in Drosophila. They showed that if you lower the insulin growth factor receptor activity in the Drosophila brain, the fly can remember longer, and it develops less so-called fly dementia at its old age. Okay, so it's protective of dementia, less insulin signaling. So this is the grand summary of my entire talk. So I talked about the supply chain logistics, and I told you this whole story about how the supply chain logistics can be managed using a combination of synaptic activity, developmental cues, and the several different vesicle recycling enzymes and kinases and phosphatases. Okay, and that's the whole intelligence, chemical intelligence of the cell. All right, and thank you very much for your very, very attentive talk. Questions? And then we introduce Professor Krishanu. He's our respected founder president who has. Here is our respected founder president. He is PhD in chemical engineer, and he has created all this universe. And he has been quite impressive. To you. What I saw today. This is my first visit. Well, yes, sir. Go ahead, please.
Dr. Krishnarao, I'm so pleased, so indebted, so grateful for your sparing time to my brilliant.
Pleasure is all mine.
To my brilliant students, faculty member, researchers. We are having 8 research scholars in this institution, and Dr. Ben Malik is a great knowledgeable person all these years in JNU, but he's really bringing a game change. I listened your whole lecture among 3 other meetings.
I'm really impressed, and thank you very much.
But I have to grasp, because when I hear 3 programs, 4 programs, then I cannot concentrate. But what you spoke, the molecular motors and all, dementia and Alzheimer, Parkinson, these are my very favorite subject. I am speaking to at least half a dozen neurologists and neuropsychologists on these topics. How can you command this? Because this is for everybody.
Yes,
And
it is. Yeah.
although my brain is still perfect because maybe after
So there
10
is a reason for it. There is a reason that some brains remain alert for a very long period of time, and
right,
the reason is some of us have this endowed gift of
right,
making a the small tripeptide called
right,
glutathione.
right,
So the glutathione is a natural antioxidant that we make in our cells, and some of us
right,
can make it much better than the others. And
right.
those who have the ability to make glutathione efficiently remains alert for a very long time.
Great. Well, I'm working 18 hours every day, and I think one says,
So
as
that's
more you
I
load
hope
your
you
brain,
continue doing that.
exactly, actually. Sir, I will come with all my student, faculty member, and researchers to your institute, NBRI.
Sure,
It
please
is
do.
very
Please do.
close to my heart.
You're
Uh,
most welcome.
Even before Abdul Kalam ji inaugurated, where I was present there, and the lady
Yeah.
was director, even before that, I was in contact. I visited also many of your faculty members were parents of my students in schools. So I will tell to VL Malik ji and Rachna Mehta and
Yeah, yeah.
others,
All of you are most welcome. Yeah.
sir, because your institution is a god of brain. I don't think any other institution in the
Yes,
country.
it is indeed. NBRC has pioneered brain research in this
Correct.
country. Indeed. Yeah.
Sir, one
And
question. Yeah, sorry, so
sure.
please.
Yeah. Yeah. Professor P.N. Tandon, I think the all credit goes to him for
I
single-handedly.
Correct. I used to do the conferences, neuroscience, and B. Tandon came two times to my conference in Europe, once in
Yeah.
Austria and one in West Germany. Sir, one
Yeah.
question to you. I believe, my faith is that we Indians, Indian scientists, nobody can supersede in this area anywhere in the world. I was 28 years in West Germany. I know all great institutions, but my faith is I want to know, am I right or am I wrong, that nobody can supersede us in the area of brain, Alzheimer, Parkinson, and all these molecular motors.
No, we do have to catch up a lot. But yes, indeed, neuroscience in India has come of age, and there is a lot of good labs now in this country,
Right,
and a lot of good students and postdocs are produced.
right.
And but we have a distance to catch up.
But we are in the
We
country
are
of
doing very well. Definitely we are doing very well.
we should, I appreciate. So, P.N. Malik ji, Rachna Mehta and all, we will go once. I will come with you to visit, sir, and
Sure, please do. Yeah,
it is
it'll be my
whatever.
pleasure and honor also.
So grateful, grateful.
Yeah.
So, Dr. P.N. Malik, I'll come again. I have to rush to the Haman now in AKC house, but I will come again to meet your students and participants. But at the moment, I just wanted to thank Ray Saab. Thank you.
We are so grateful, sir.
Thank you, sir.
Thank you. Yeah.
Great, great. So, good to talk with you.
Yes, now Rachna, continue with the question.
We can now take up the questions. Yeah, please.
Good afternoon, sir.
Sit down, sit down. You don't have to stand up.
I had two questions. First question was that we have these transporter molecules for both anterograde and retrograde transport. So for each of the package or the parcels or the molecules which have to be transported, are there equal amount of transporters? Like,
Very good questions. So the question is, whether the number of motors are equal to the number of cargoes. Answer is no. So the cell has only few types of motor. I showed you 3 types of motor to transport all these, tens of thousands of different variety. So obviously there is a shortage of motor than the cargo. Also, numerically the number of motors are not same as the number of cargoes. Obvious, because if you have so much motor, it will itself create a traffic jam. So the cell has to make a very optimum utilization of the number of trucks that it has. This same as this, like a transport operator has a few trucks, but using a few truck, a transport operator can manage to supply a lot of different goods. Same way the cell manages supplying a lot of different cargoes using a few few motors in that cell. I don't know exactly the ratio, so I won't be able to commit a specific number, but it's a very good question.
I had just one short question. Like you said in EEG, while measuring, we aren't able to localize, like, we get a generalized view, but not able to localize the
Right.
area.
Yeah.
So, like, if suppose, like, does the size of the brain matter? Like, suppose if we use some animal which has a bigger size brain, if we use a sharp electrode.
Well, we have the biggest size brain, unfortunately. But yes, so if you have a smaller animal like primates, you can do it little better. And even smaller animal like Drosophila, you don't need EEG, you can put an electrode into it. That's where the model comes into play, right? I can't drill a hole and put a chip in it, although we are doing it now. So only in special cases when the ethics approve, the Neuralink chip can be inserted in your brain, or if you approve yourself. But otherwise, none of you here will be happy if I drill a hole and put an electrode inside, poke it inside your brain. So that is the reason why we do it in a model system. So monkey doesn't. We can actually drill a hole and put an electrode and measure. But to put the electrode, we need to have a gross idea where to put the electrode, and that comes from the study in the human brain. So here the size is an advantage because the brain is bigger. So modeling signal local source localization becomes easier because the signal strength is stronger. So those things are, sometimes big has an advantage.
Okay, thank you.
Yeah.
Other questions?
Thank you for a wonderful talk, sir. I have a question about when you are talking about the moving of these vesicles in presence of insulin. So, does these, if we are treating them with the presence of insulin, does they become insulin dependent? Or we have some alternatives that we can induce these vesicles to move faster or slower? What kind of control that we can offer?
So, see, insulin is signaling through the VPS34 here in this case. So, if you stimulate VPS34, then you won't need insulin. And there are many other signaling systems that can also stimulate VPS34. As of now, we really don't know, what is this connection. So we feel that, this particular network is specific to insulin. And although VPS34 is, at the base of many other signaling network, those will not stimulate the recruitment of KIP16B. So how does it happen is still, not very clear. So for this purpose, we developed another molecular, fishing hook. So it is called split apex assay. So what we did was we split this plant peroxidase called apex into two parts. Now, when you break it into two parts, it will lose the enzyme activity. And then we put these two parts onto two different proteins. When these two proteins come close to each other, like freight, these two part of the apex will get together and constitute the apex enzyme. So when it constitutes this enzyme, peroxidase activity will generate oxygen free radical in presence of H2O2. And that can actually allow a biotin free radical, phenoxybiotin radical to form, and that can then attach biotin molecule to neighboring proteins. And it was shown that this effect, the oxygen free radical, can only diffuse up to 20 nanometer. Beyond 20 nanometer, it gets quenched, because there are several other water molecule inside the cell which will grab this oxygen free radical. So therefore, it uses as a good proximity labeling tool by which you can label the proteins which are in the neighborhood. So we did that experiment and we fished out some 4,000 candidates. Now we have narrowed it down to a few hundred, and we are still struggling to figure out the connection from the, unfortunately did not connect to the insulin receptor. So we still don't know, but it will tell us how VPS34 is recruited on this vesicle.
Okay, thank you, sir.
Sir, I'm curious to know, like, what is the relation of the rhythms of the brain with axonal transport? Like,
Again, another very good question. Well, again, I don't know the answer to this question, but the brain rhythms that you see are result of the synaptic activity. And I showed you some data which shows that synaptic activity is required for maintaining the axonal transport of certain type of cargos. So this is a, again, we did this, but we did not somehow complete that experiment. So we found that the synaptic activity has differential impact on different kind of transport. So for example, cholinesterase transferase transport and RAF4 transport are differently affected by the same activity. So if we use hemicolinium, it inhibits cholinesterase transferase transport, but it doesn't inhibit the RAF4 transport. Whereas if we activate the, if we inhibit acetylcholinesterase, which will activate the synapses, it will affect RAF4 movement. It will affect the RAF4 movement, and it will not affect so much as the cholinesterase transferase movement. So in a way, this brain rhythm will affect the transport. How is something is very complex, and we need to refine this system, and this is just the beginning. I haven't seen too many studies in this area so far, so it's just the beginning. So I'm sure you will read much more in the future. Okay, yeah, sure.
Hello, sir. Excellent talk. And I was just wondering that you told dynenin shows movement opposite to the kinesin. So how does kinesin takes care of this? Because dynenin can become the roadblocks. And another question is, what happens to this transport when we sleep?
Okay, so another very good question. So yes, so this is a very difficult problem, because two motors walking on opposite direction will collide with each other, and why doesn't they collide, right? Okay, so this experiment was done, and it was seen that dynein has this intelligence to switch track when it comes to an obstacle. So when a dynein and kinesin cargo is facing head on, the dynein will just give way. It will just switch track, and the kinesin will move, yeah, like a Rajdhani Express. So, and this is because dynein is a very large beast. It has a long leg, so it can switch. But kinesin has a very short leg. It's like your leg tied to your knees. So you are only allowed to walk on a very narrow straight. So this is one. The second is that more often than not, a kinesin motor and a dynein motor are attached together. Intrinsically they are attached together. And there are kinases and phosphatases. They have opposing effect on this motor. For example, a phosphorylation of kinesin by one particular kinase and a phosphorylation of dynein by the same kinase will produce opposite effect. It will activate kinesin and it will inactivate dynein. So there is this intrinsic trade between these two motors. When one is active, the other is inactive. And all dynein has to ride kinesin to reach the end of the synapse, end of the axon, because dynein is a minus n directed motor, and the roads in axon are all unidirectional. So dynein ride on kinesin to reach synapse, and then kinesin ride back on dynein to come back. And this is how this, so this is dual engine, train, which keeps moving back and forth. And the best example is the melanosomes in melanocyte. The chameleon, you've all seen how it changes color. So the chameleon changes color by moving these granules called melanosome, which scatter light. So this is not a real color, not a chemical color. This is just a scattering of light that changes the reflection of light from the surface of the skin. And these granules move on this dynein kinesin pod. And depending on the hormone, either melatonin, melanin, or other, this movement can be regulated. The dynein can move it to the center of the cell, or kinesin can move it to the periphery of the cell. It also happens on our skin. You go out on the sun, you develop a dark tan. And that is because these melanosomes are distributed on the kinesin. Kinesins are active and melanosomes are distributed. You stay indoor for several days, you become fair, because all the dynein is active and these granules come close to the nucleus and the cell becomes transparent. So this is how it happens in the neuron also. The kinesin and dynein are always in tango with each other. And that's how they don't collide with each other. But they do collide occasionally. I have examples of those also in my experiment. What happens during sleep? In what context?
What happens to this transport?
Oh, what happens to the transport during sleep? The transport goes on. It's an intrinsic property of the cell. It doesn't stop. In the sleep also it doesn't stop, but the flux may vary. Again, I don't have an answer to this question. I can't open an animal while it is sleeping.
Any question?
So suppose like we are exercising, so we need more acetylcholine in our muscles. So at that time, like the transport would increase, right? Because like
Hopefully. Yeah, I haven't done that experiment, but
Okay.
hopefully.
so my
Yeah.
question is the regulation, right? With increase in demand, the speed and the amount
Yeah,
of
yeah, beautiful
transport.
question. So I have some inkling. I don't have a data, so I won't tell. I can't give you a definitive answer. But the my hypothesis is that this kinesin cholinesterol transferase interaction is dependent on the pH of the environment, and the pH of the environment will change when an action potential travels through that area because of the change in this flux of the ion. The pH and the potassium flux will change, and that could actually regulate the interaction between the cargo and the motor. How, I don't know, and I hope I get somebody who is curious to answer that question to do this experiment. But we did somewhat similar experiment by using a cation channel, photoactivable cation channel. So when we activated this cation channel and pumped more cation into the axon, it stopped the movement of the RAF4 vesicle. And the RAF4 vesicle movement are antagonistic to the CHA movement. So hopefully the CHA will be more when the RAF4 is less. But again, and I don't have a clear answer. Okay, so thank you very much.
So we would like to thank Professor Ray for such an informative talk. And now I would request Dr. Rachna Mehta to felicitate sir. Moving forward, amongst the participants, we have three groups. So Rishika from group 1, Arpita from group 2, and Huma from group 3 are requested to step outside and provide your valuable feedbacks and insights to all of us.
So actually, we had made three groups for the participants who had been doing their lab work in rotation. So any one of you, as you volunteer to present the feedbacks, you can please come over and in the sequence, like from group 1, group 2, group 3, can please give your feedbacks.
Good afternoon to everyone. I'm Rishika Dhapola. I'm from Central University of Punjab. Currently, I'm a PhD scholar working in finding the treatment for Alzheimer's disease. So, first of all, I would like to thank the organizing committee for organizing such a wonderful workshop. This was so insightful, and we got to learn a lot from this. So, starting with the lectures, there were very much to learn about the genetic association and with the learning disabilities, neurodegenerative disease models, and management of those diseases and recent advancement in those diseases.
So after that, we got to have the hands-on training on various experimental techniques, including behavioral, molecular, and biochemical studies too. Each one of us got the opportunity to work by ourselves on those experiments, and we got to learn a lot from that. And after that, we got to learn about how to correlate our wet lab data with the mat lab. We learned about the in silico studies too. And there were industry experts too who told us about the various softwares we use in our experiments, like animes, and the techniques we use, like Western blot. So this was a very insightful workshop. And yeah, that's all. That was very insightful, and thank you so much for this workshop. Thank you.
Respected Founder President Dr. Ashok Kumar Chauhan, respected Professor and Director Mian Malik sir, organizer Dr. Rachna Mehta ma'am, and all participants. My name is Arpita, and my group members are Mr. Sandeep, Suraj, Ikra, Anuradha, Himansi, Sachin, and Dr. Praveen Kumar. We found ourselves so comfortable to learn here. During these 5 days, we did not feel like we are away from our university or our teachers, our friends, or our supervisor. Here, we feel very comfortable in environment to study, to learn. All teachers, professors, volunteers, they were very friendly to us and guided us whatever we ask, and whenever we need, they teaches us very kindly. So we feel here very lucky to be here in this workshop. And Professor Brian Malik sir heads, they always say to sit in front and actively participate. It really inspired us, and thank you so much.
Arpita, from which university you are? Arpita, here is founder. From which university you are?
Sir, CMP Degree College, University of Allahabad.
You will have a big career. The way you expressed everything, behavioral science is 100 out of 100.
Thank you, sir.
And keep touch with us.
Good afternoon, everyone. I'm Dr. Huma from Jamia Hamdard. Recently, I'm working on ADHD. Thank you so much for MET University and the Department of Neuroscience. Special thanks to founder sir and director and professor Malik sir and organizing committee member Dr. Rachna ma'am. Thank you so much, and all other respected faculty members and students. So this workshop help us to attribute new aspects and boost our mindset and working practice. All sessions have been very productive, and we learned a lot from this neuro workshop. Personally, I believe this kind of ANIN workshop is excellent for anyone attempting or trying to conduct some molecular, behavioral, and computational spatial research. It offers a perceptive and yet concise scenario. Thank you.
Great.
Yes, sir.
No, I think it's freeze. It's freeze. Okay. Anybody else wants to share with you?
Moving ahead, I would now request Dr. Rachna Mehta, the organizing secretary of the workshop, to please present the workshop report.
Very Honorable Founder President, sir, dignitaries, participants, and friends, a very good afternoon to all of you. It's my pleasure, and indeed I'm delighted to present this report on the 5-day workshop that was conducted under the chairmanship of Professor B.N. Malik, Neuro-behavioural Studies using vertebrate and invertebrate model organisms. So this workshop, the theme was to understand behavior regulation by the brain through the changes in the cell, molecules, and neurotransmitters. And this was an initiative that was partially funded by Science and Engineering Research Board, Department of Science and Technology under the Scientific Social Responsibility head. The objectives of the workshop were to develop critical thinking skills, to formulate scientific questions, nurture interest about the brain, and definitely for the academic collaborations. Few insights into the workshop. It was planned to provide hands-on training and understanding the correlation of the molecule to behavior research in rodents, where you had the opportunity to see the behavior changes in the rat, mice, zebrafish, C. elegans, a versatile model. You had also done the biochemistry experiment, molecular and cell biology, starting from the protein extraction from the brain isolation and the SDS page running, and also the demonstration by the experts from the company. Systems neurosciences, which provided you insights about the mathematical modeling and computational neuroscience. And we also had the demonstration of the stereotaxic brain surgery and histology to approach the specific regions of the brain for chronic studies using staining and immunohistochemistry. We had resource persons from AIMS, New Delhi, IIT, IGIB, Jamia Hamdard, JNU, NBRC, Naipur Mohali, Punjab University Chandigarh, Ashoka University Sonipat, and NII New Delhi. The participants, which spans 3 of the assistant professors and 22 PhD students, which were from Central University of Punjab, ACBR, Delhi University, AIMS, CSIR Institute of Microbial Technology, Banaras Hindu University, University of Allahabad, Jamia Hamdard, Aligarh Muslim University, Baba Masnath University, Rohtak, Swami Vivekanand Subrati University, Sri Guru Gobind Singh University, Tripura, and Amity University, Uttar Pradesh. We started with the inaugural program, which was held on 18th of March, and we were fortunate enough to receive from the very first day the blessings of our Honorable Founder President and the guidance of our Vice Chancellor, Madam, support of Dr. W. Silvamurthy, sir, and guidance from Professor B.C. Das, who were also present during the inaugural program to encourage us all. We had eminent speakers from the very first day, where we had Professor Shashank Sharat Kale, neurosurgeon and expert and eminent neurosurgeon known across the globe for his contributions and understanding the brain lesions, effects, and the new strategies for its management. We also had on the inaugural day Professor Subrato Sinha from NBRC and ex-director of NBRC, also the National Science Chair, SERB DST, who gave us insights about the biological basis of learning disability and how to reveal genetic associations to the learning disability. We had several interactive and participating sessions where all were encouraged to ask questions and interact to the fullest to make this inaugural day as a success one. Day 2, we were fortunate to have Dr. Binu Kumar from IGIB New Delhi, who discussed about the several signaling pathways, exploring the influence of the cyclin-dependent kinases on the neuronal death, health, neurodegeneration, and behavior impairments. Professor Dulal Panda, who is the director of Naipur Mohali, he gave us insights about the tau abnormalities in neurological disorders and discussed several potential therapeutic strategies. Professor Rajat Sandhi from Punjab Universities gave us insights about the Alzheimer's disease and how we can use the zebrafish as a model organism for the different neurobehavioral studies. We also had talk from the industry experts, that Dr. Sandeep Majumdar, who is a country manager for Gentech Private Limited, who showed us the applications of using the behavior software in rodents, zebrafish, and C. elegans, and how to use electrophysiology and its applications. Day 3, we had talk from Professor Suman Jain, who is an expert of transcranial magnetic stimulation from AIMS New Delhi, who showed us neuromodulatory role of the external magnetic field stimulation in the rat model in the different neurodegenerative disorders. She also showed us the applications of the transcranial magnetic stimulation. Dr. Sushil Kumar Jha, who came from JNU New Delhi, gave us the insights into the neural plasticity and the compensatory mechanisms of the brain. Professor Sohail Parvez from Jamia Hamdard, he working on a span of models starting from invertebrate to vertebrate. Was really exciting to listen to him and understanding the neural basis of cognition. Dr. Sunil Kumar Khatri, who is the Dean of Dean Research at Amity University, Uttar Pradesh, took us through the journey of how Amity University is having a platform for the research and innovation opportunities. We also had Dr. Smita Sahu during the same day, who gave us insights about the patents, copyrights, and such, where the students can also interact and approach her anytime as required. Day 4, we were fortunate to have Professor Pankaj Seth from NBRC New Delhi, who gave insights about the isolation and characterization of the human brain cells and how to use these cells in case of viruses affecting the brain functions. Dr. Krishna Milnatur, who came from Ashoka University, showed us the evolution of sleep, the effects of the sleep on brain plasticity. We also had Dr. Chetna Sachidanandan from IGIB New Delhi showing us the versatile zebrafish model for the human neurodevelopmental and neuropsychiatric disorders. So today, day 5, we were fortunate to have in the morning Professor Chinmoy from IIT Delhi, and followed we had Dr. Arnab from NII, and now we have Professor Krishanu Ray. We were really grateful to you, sir, for your talks on day 5. Students have, and all the participants had done behavior experiments where the key highlights were the spatial learning and the motor coordination that they have done in the control and the disease rat model. In C. elegans, chemotaxis and learning behavior was shown, and the students were really very excited to see the social interaction and the isolation behavior tests in case of the zebrafish, and also going through the different developmental stages. They were also demonstrated the stereotaxic brain surgery and histology, how to do the cryosectioning and the stereotaxic coordinates to reach the different brain areas and doing the brain surgery. In biochemistry, they have done the isolation of the proteins from the rat brain, and SDS page with demonstration from the industry experts who showed them the recent technological updates. They have also done in systems neuroscience, the functional characterization of genes, identification of microRNA and drug as regulators, and acquisition and monitoring of the brain activity using EEG machines, signal processing using MATLAB and Python. So I hope you all had an enriching experience here at AINN AUUP, and we hope that your learning here will take you to new heights, and definitely we look forward to meet you again at a new platform. Thank you very much for being here and being a part of this workshop. Thank you.
Yes. So now, before we distribute the certificate, we are very glad and fortunate that the founder president, sir, has kindly agreed and spared some time to bless us all with his speech. And then we will go for the certificate distribution. And I am grateful to Professor Ray, who has kindly spared time to give the certificates. Okay, and then we will break for lunch. Yes, sir, please, your blessings.
I must tell to all my student, participant, researcher, but keep it confidential. This is the most beloved, respected institute at the campus, because I asked the question to most respected Krishna Raoji, and the question was that can we in India become number one in these areas? And he replied very well that we should do still further work. I agree with it, but I must tell to all my participants for workshop, from now onwards, you are also member of the Amity Parivar. I was listening all 5 days in between whenever I find time. I'm really proud of each one of you who has joined from outside, and 3 of my participants in the workshop. I go on getting reports minutely from the campus of the whole day. I have got an intelligence and vigilance wing. They send me in the evening minute-to-minute report, and they were giving very good report from this workshop participants. Sir, they are brilliant, interested, devoted. So keep touch with us because you are from good universities. अच्छी यूनिवर्सिटी से हो पर हमारे से संबंध बना के वन से synergy is a great weapon like Brahmastra. The best of best between two companies. Your institutions are great, your universities are great, but be in touch with us. I must further tell how much interested my family is. The eldest daughter of our Chancellor Atul Chauhan ji, Soumya, is studying in epigenetics. It took a long time to understand what is epigenetics. You must be knowing it better. And she has been admitted in Rice University, which is #1 in the world in Houston, in USA. And her younger sister Swani wants to study, will be going 12th this year in the cognitive sciences. And she told me there are two areas, biological and psychological. So if anybody wants to go abroad, your university will be helping, but I will also fully help you in all means and possibilities if you want to study abroad to see what they teach, what they learn. That is why I sent Soumya and Swani to see what best we can do, what more we can do, as Krishna Rao ji told. So you are in a branch. I must compliment your parents, your advisors, your family member, and you that you selected this area. I think this is the best area where one can do any reach to any success you want. So my advice to you is be in touch with us. I will give you, I'll write you also my letter to you, by email to you, so that you know on which email or WhatsApp you can directly contact me, because here thousands of people come, but you are the closest to my heart. This is for my trainees, those who have taken part. My dear students, I'm very proud of you. And Dr. B.N. Malik is a great scientist, a great researcher. When you see in which societies he's president, he has been awarded, there are half a dozen or more accolades he founded, and we are proud for his scientific arena. Also, Rachna Mehta, before Malik ji joined, you were the officiating head of AINN, and what did wonderful control even before having a official head. So I must compliment you, Rachna Mehta, and we always think of our Malika Chatterjee, who has been given a fellowship to go to Boston and do there. I think my policy is best of the East and best of the West. And if you see best of the East and best of the West, then we can be superb. Nobody can supersede you. So all my 9 faculty members, they are really great. I will try to come more often to you, and we will all go to NBRI. I will take everybody with me, and we'll spend 3-4 hours there. Krishna Rao ji has very kindly told that he will receive us, and I want to see BNRI has a lot of prospects, a lot of possibilities for us. So this is at the moment. Be in touch with us. You can directly write to everybody, because I want to bring revolution through you in the world, not in the country. Through you, and you are capable, willing, devoted, sincere. I will not say it if I am not. अगर मुझे विश्वास नहीं हुआ, ये सारी क्वालिटीज जो मेरे पार्टिसिपेंट थे, 25, 23 from us and rest from outside, and my student faculty member, each is a jewel. So I am with you. Make a big, when you go from here, make a big aim, write on a paper after listening to founder president. Now this is our aim of career. If you don't achieve it, it's my failure. It's my failure. So please be in touch with us. Ian Maliki, thank you very much. I have to rush to the Havan now again. We are having Diwali Havan here. My blessings to everybody. Good wishes to everybody. Thank you very much.
Now, with this blessing from founder president, we are very, very lucky that our dean could spare some time, Professor Das. So it cannot end without that. So, Professor Das, a few words from you and then the certificate distribution.
Good afternoon, one and all. And most respected Honorable Founder President, sir, and you have just listening to him. It's a great occasion that you could see him and he blessed you when you have completed the 5 days workshop and you will be leaving today. It's a great occasion that you could come, those who are the participants, 25 participants, and yeah, 24 participants. You have come from many places, different places in Delhi NCR and outside. And I think you had a great journey, the great learning rather, these 5 days. And as I told in the inauguration day that so many very chosen speaker in the different areas of neurosciences and neuropsychology, and which started with none other than the Professor S.S. Kale. He is a clinician, known clinicians, the neurosurgeon, and Professor Subrato Sina, which was the inaugural day lectures, and then concluding by none other than Kisanu Ray. He is the chair and professor and chair of the biological sciences in TIFR, Tata Institute of Fundamental Research, is one of the top research institutes in the country. And now he is the present director of NBRC. Everybody knows those who are in this specialization, NBRC. So he is the new director. I wish he will bring a new things in the neurosciences here, and it's a very good institute. We know it, how it's originated, and one of the excellent institute in the country. And you had the opportunity to listen to him. So there are, I will not take names, but you know already there are people from the NII and also from the Suman Jain and then Susil Jha and Suel Parvez from Hamdard. Then Pankaj Seth, I met him, I think yesterday only. Yeah. So I know him very well. He was member of this EIN committees, several committees. And Chetana Sachidanandan and Chinmay De and Arnab Mukhopadhyay, and today is Dr. Ray. So it's a great journey, and they are excellent researcher, have a huge scientific standing in this field, and you could listen to them. I am sure you will carry a lot of message, lot of science, lot of, you had the scientific feast, I will say, and that you had the feast on the first day, the dinner, but then you will have the scientific feast, and you will carry some new knowledge with you, and you will know that what are the model systems. We know it. There are, starting from today, it's ended with the Drosophila, the C. elegans, and rats and mice and all. There are several model systems are there, and that gives us an opportunity to work in a better way to understand the neurosciences, how it correlates with the human behavior, because we extrapolate the data to human. That is the main aim or the ultimate aim. And these systems we use because of the convenience. The system is simple. Human system is the most complicated and evolved system in this on this earth. So we take some simple one cellular, single cell and the organisms, or some Drosophila like, which has a life cycle is very quick. So these things helps us to know how it is, the behavior is changes, and there are, you know, the Obed Siddiqui, he worked with the Samir Benjar, who got this behavioral study, the Nobel Prize. So this is how, and he was the director of this TFR, or this biological sciences attached with the TFR, the NCBS. So he was the first director, and he worked with Samir Benjar, who got the Nobel Prize for the behavior. So I will not go into the more details, and but then these systems helps a lot, and that is the title of your workshop was the Neuro-behavioural Studies using vertebrate and invertebrate laboratory models organisms. So this is a great opportunity to know how you can utilize other animals and to easily interpret the data, and which is you can do much easier way than your, I'll call you back later. So easier, but then this the after all good things, the end comes one day. So you have to leave this conference and the MIT campus. I am sure during this 5 day stay or 6 day stay, you must have seen some departments or gone around in the evening, that if you had time or if you are in the Delhi NCR, you must have immediately to catch the metro or something. But you should find out something even today also when you leave. Before that, at least go around the campus and know what all is being done and what are the departments, neuroscience and other departments, also related departments, so which you can see. And nowadays, any science you do, and neuroscience comes under health and allied sciences. But it is now the age of digital age, and digital health is very important. And neuroscience is no exception, that artificial intelligence and the digital health system or digital system of data analysis and analytics and all can help a lot in doing great science or the interpretations and their evaluations. So we have recently have established with huge spend of money. Dr. M.K. Datta is the director of that institute. He was here in an engineering department, but he has it by the computer science and all. So he is now the director of that, and it has several crores have been spent to put a huge the computer system here. It is the best in the country rather, and it is also being improved or the further models. So this is the Center for Artificial Intelligence is recently established, and if you have time to look at say E2, E3 block, I think E3 block. So you can go to that and see that how it is. It is excellent. And there are several projects have been also being immediately moved. There are others are also working on biotechnology in my department, MIT molecular medicine and stem cell research. So this there we have already written a 1 crore projects, and there is 8 crore projects on two different projects, ICMR and Ministry of Education, on artificial intelligence. So it has a immense importance and help. What you can do in a regular way, if you use the artificial intelligence, it is reduce the time at least 30 to 40 to 50% even the time consuming, which we can interpret and we can know. As you know from the internet, the Android phones, when you see something and again you go to that, it automatically you write one word, it appears immediately. So these are simple example of the artificial intelligence, that how it can be utilized. So this has a great opportunity to utilize that artificial intelligence system and the including the modern system of the modern methods. So using these models, you can use the genome editing and all the CRISPR-Cas and many methods have come out, and the NGS, the next generation sequencing, which can be utilized. Any advanced technique can be utilized, including the standard and very old but gold techniques that the neurochemistry and all can be utilized, but they can be further interpreted with the new technology to give a new insight on this and to interpret the behavioral aspects or any other neurological aspect. I think brain is the one organ which is least understood. And as you can go on doing your whole life working on this, you can spend whole life, but you cannot end it. Your small aspects you want to learn it. and it is directly associated with the human benefit or societal benefit. So what you describe, it is will be translated into the human benefit. So, like just now I was coming, we are discussing who is the best person to glioma, the brain cancer, the one of the dreaded brain cancer, that and how who is the best to treat it. It is there are treatment going on, it is not responding. So we do not understand much of that. So there are huge number of things, including behavior and any type of disease, and particularly the sleep. The original sleep person is here, your head, Professor Malik. So he has spent his whole life. Still, it is how much is understood. So why you get sleep, you do not get sleep, what happens, and all this. There are huge number of huge understanding is needed. Lot of things to be discovered, waiting for the discovered. Lot of Nobel Prize is waiting to be given, but you have to develop the really what I say is the love with your subject. You see, what your department gets, your promotion and all these things, it's least matter. But if you are good, if you can contribute something which can come to the human benefit or some behavior disease or any type of disease, neurological diseases, so that you look for. So this is should be a fashion or a passion for your subject, that it is my career and this is my passion to do on neuroscience, and since you have taken, this is your career. So what you do that reflects in your other things. You would say the promotion and then all these things, position, that comes automatically. If you are good, if not today, tomorrow, or not tomorrow, day after tomorrow, people will recognize you. Your work cannot go waste. So this is what is the most important thing, to develop love with your subject. I know anybody who has come to this stage of director or chair or all big scientists, got all the award, they had faced a lot of trouble or difficulties in their journey, and they have come to this stage. And this is with the work only. So nothing to be compromised with your work, the contribution to your own specializations. You are not doing anybody's specialization. So I will not take much time, and it is, I think it's a lunch time, and you are all hungry, and there are other programs are there. So thank you very much. Carry the message from here, what the esteemed speaker has spoken, the knowledge, and I, on behalf of the Amity University and our Honorable Chancellor, Honorable Vice Chancellor, and our colleagues, the professors, deans, and all the faculty and the students, every fraternity of the Amity, I wish you all the best in your career, and you are doing good, and I wish you will do good and you will come up to the expectation of your organization, of the country, and contribute to the nation building. Thank you very much, and all this best.
Okay, thank you, Professor Das. It is so kind of you to spare some time and speak. Okay, so now what we will quickly do, because just now Professor Ray tells me that he got a message from his office and he has to travel to NBRC 50 plus kilometers. Because see, this is all of you as a student know, understand, March ending is something very crucial. Therefore, even Arnab and Chinmoy couldn't stay back, and he also has to go, and he spared some time. So what I will request Professor Das and Professor Ray, and Professor Ray has kindly agreed to quickly that they will be here to distribute the certificate. That might take about 5 minutes, and then quickly we'll start the lunch, and Professor Ray and Professor Das will honor us, and then we will leave him. Okay, so please, yeah, please. Yeah, please. But then you have to be, because he start the certificate part. And, go ahead quickly, because everything is in a rush, and particularly March ending and everything. Yes, one by one. Yes,
Now the certificate distribution session is going to start. The first participant is Miss Rishika Thapola. Miss Nilima. Mr. Dheeraj. Miss Sandhya. Mr. Arpit. Miss Mehak. Mr. Sandeep. Mr. Suraj. Mr. Ikra. Miss Arpita. Miss Anuradha. Miss Himanshi. Dr. Nilanjana. Mr. Sachin. Mohammad Anas. Miss Huma. Miss Navneeta. Dr. Surjit. And Miss Garima.
So can I request our AIN faculty and students just to have a group picture? Come. Yeah. Whatever the participate तो हो गया ना? हो गया के साथ हो गया everybody. AIN party faculty and students of faculty. So you can come with the faculty can come also. हाँ तो आ जाए. For then students भी है. हाँ चलो ठीक है. अरे आ जा quick so that they have to leave. So after this, Anubhuti will give a vote of thanks. अरे तुम लोग आ जाओ. Actually, you can come on this. ठीक है एक ग्रुप फोटो ले लीजिए सबका. वॉलंटियर भी आ जाओ. तुम लोग बाहर क्यों है? जल्दी आ जाओ. सबका चेहरा याद रखना है ना. Will remain in the memory. You can come in front of. I don't mind. Yeah, come on, grow in the. जहाँ भी करना है. हाँ. चलो आ जाओ तुम लोग जल्दी बोलो आ जाओ. क्या नहीं जो नीचे बैठ रहे हैं उनके पनिशमेंट हो जाएगा. Okay, now take the picture. Group picture अच्छी तरह से आना चाहिए. एक सामने देखिए. हाँ. Thank Thank you so much. Okay. So now vote of thanks because that is without that things cannot end. And then Rishikesh. Participants, if you want a group.
Now I'd request Dr. Anubhuti Dikshit, Assistant Professor AINN, to take the stage and address the vote of thanks.
Thank you, Priyanshi.
Because I have to do something, he has to go away. Yeah.
Good afternoon, all. It's my privilege to have been asked to propose a vote of thanks to on this special occasion of valedictory session of 5-day workshop on neurobehavioral studies using vertebrate and invertebrate laboratory model organisms. On behalf of Amity Institute of Neuropsychology and Neurosciences, Program Director and Head Professor B.N. Malik, Workshop Organizing Secretary Dr. Rachna Mehta, and all committee members, I thank our esteemed speakers for taking their precious time and enriching our brain with their scientific deliberations. Their knowledge and expertise in the field of neuroscience have put India in forefront, and I'm sure that after listening to their lecture, all of us are encouraged to develop innovative ideas and solutions for the neuroscience research. We take this opportunity to thank our Honorable Founder President Dr. Ashok K. Chauhan sir for his kind blessings, guidance, and encouragement to conduct this workshop. A sincere token of gratitude to our respected Chancellor Dr. Atul Chauhan and Vice Chancellor Professor Dr. Bhavendra Shukla for their continued guidance and support. We are very thankful to Dr. Silvamurthy, President STIP, who is like a mentor for us. He has been there for us and never fails to appreciate and encourage on our achievement, whether it is big or small. We wish to express our deep gratitude to Professor B.C. Das for guiding us and enlightening our path as Dean Health and Allied Sciences. We thank all esteemed advisors, deans, and heads of institute who have honored us by attending this workshop. We greatly appreciate the help extended to us by Wing Commander M.K. Sharma, Dr. J.S. Sodhi, Major General J.P. Singh, IT support, and their entire staff for their enormous cooperation in this organization of the event. We would like to thank all the participants for choosing to be a part of this workshop. Their enthusiasm and active participation has been a key to the success of this workshop. An event like this cannot happen overnight. The wheels start rolling weeks ago. The workshop would have not been possible without the tireless effort, continued guidance, and mentorship of our director, Professor B.N. Malik, hard work of Dr. Rachna Mehta, and definitely all the AINN faculty members who have been playing important role in organizing this event. A special thanks to AINN support staff and student volunteers for constantly working towards organizing this event. We hope that this workshop has been a stimulating and learning experience for you, and we look forward to interact with you in coming days. Thank you once again.
Thank you. Thank you, ma'am. Now, each and every participant, the dignitaries, and the present faculties are requested to head for lunch. As we conclude these insightful 5 days of our workshop, we want to extend our heartfelt gratitude to each and every one of you for your active participation, engaging discussions, and valuable contributions. These 5 days have been a journey of discovery, collaboration, and growth as we have delved deep into the realms of knowledge and expertise. Let's carry forward this momentum and continue to foster a great scientific discoveries as well as collaborations that are to be coming. Thank you so much.